Hello, Bachas. Welcome to video solutions of your part test. So today again in this video, we are going to discuss about your botany solutions of section A and section B. Okay, so let us just dive into the topic. But I'm thinking before diving, let me tell you one small story about myself. So hello, Bachu. I am your botany subject matter expert at MEMONEET Ronia. So I'm currently a final year medical student at Government Medical College, Mehboob. So today in this video, as usual, we are going to discuss about solutions of section A and section B of botany in detail. Okay. And if you have any doubts, do put them in comments below. I'm happy to solve them. Okay. So today, before starting with solutions, I want to tell you a little story about myself. तो जब मैं नीट लिख रही थी in 2019, nearly 2 days before NEET, when I had to do revision, like especially the revision for biology, that is botany and zoology, I was horribly nervous because I had so many textbooks, 2 textbooks. At that time, even biology textbook of our 11th and 12th class seems a big one, isn't it? अभी तो छोटा बहुत छोटा बुक लगता है वो ऐसा लगता है कि कितना ही छोटा बुक है एमबीबीएस के बुक्स देख के बट उस समय पे वो दोनों 11th और 12th के बायोलॉजी टेक्स्ट बुक्स भी बहुत बड़े लगते थे एंड आई वाज 2 डेज अवे फ्रॉम नीट और मुझे पूरा बायोलॉजी रिवाइज करना था बिकॉज फिजिक्स और केमिस्ट्री इज अ कॉन्सेप्ट उसमें ना कॉन्सेप्ट चाहिए होता है इंसान को अगर तुम्हारे पास कॉन्सेप्ट है तुम रिवाइज नहीं भी करोगे तो ठीक है चलो इनऑर्गेनिक में रिवीजन चाहिए होता है बट अगर तुम्हारे पास कॉन्सेप्ट है और तुम रिवाइज नहीं भी कर रहे हो तो ठीक है दैट इज ओके चलेगा बट बायोलॉजी ऐसा सब्जेक्ट है ना कि लास्ट 1 मंथ ऑफ नीट में अगर तुमने उसको रिवाइज नहीं किया है ना तो वो पूरा वॉलेटाइल 1 सेकंड में तुम्हारे दिमाग से निकल सकता है वो जब एग्जाम में तुम बैठोगे तो मेरे को इतना रिवीजन करना था एग्जाम में आई हैव टू डू सो मच रिवीजन एंड आई हैड टू थिक टेक्स्ट बुक्स एंड आई वाज थिंकिंग अब क्या करूं इतना नर्वस थी आई वाज सो नर्वस दैट आई स्टार्टेड क्राइंग इन फ्रंट ऑफ माय मॉम suddenly no nothing was happening my mom just gave me breakfast and I suddenly started crying. I was like, now what will I do? I have to revise so much. I do not have time. I was so nervous. I did not have any confidence at that time. So then I called up my friend. Okay. And she was like, I am watching video solutions and solving टेस्ट सीरीज तो व्हाट डिड आई डू आई स्टार्टेड वाचिंग वीडियो सॉल्यूशंस तो उसमें क्या होता था ना मेरे साथ आई यूज्ड टू गेट द क्वेश्चन आल्सो एंड आंसर आल्सो एंड बिकॉज आई यूज्ड टू वॉच वीडियो सॉल्यूशंस ऑफ पार्ट टेस्ट एंड फुल टेस्ट तो मेरे साथ क्या होता था हर एक चैप्टर रिवाइज होता ही जा रहा था अब फिर मेरे को वो पूरी बुक पढ़ने की वापस जरूरत नहीं है बिकॉज वो वीडियो में एवरी चैप्टर से कुछ ना कुछ क्वेश्चन है और वो क्वेश्चन में द मैम यूज टू एक्सप्लेन एवरीथिंग रिलेटेड टू दैट क्वेश्चन तो मेरा वो क्वेश्चन भी कवर हो जाता था उसका रिलेटेड सिलेबस भी कवर हो जाता था तो ऐसे 2 days बिफोर नीट आई वॉच इट सो मेनी वीडियो सॉल्यूशंस एंड दैट इज हाउ आई रिवाइज्ड बायोलॉजी that was the main reason I got good score in biology in 2019. और 2019 में तो there was lot of inflation in rank also. In 2018, just marks पे one used to get 18,000 rank, person people got 30,000 rank at the same mark. So happens if you are nervous, if you don't feel confident, you're feeling that मेरे से नहीं होगा, then don't worry. This happens with everyone before NEET. It's normal thing. Just be there. Just go with the flow. Hang in there. You will be at the other end of the NEET 2024. Okay, so just be there, be strong. I know it feels impossible, but believe me, it is not. All the hard work you have done throughout the year will be useful on the day of NEET. Okay, it will be useful on that day. Don't worry. So with that note, let us continue learning about botany. So let us dive into it. The plant parts which consist of two generations are one within the other pollen grain inside the anther, germinated pollen grain with two male gametes, seed inside the fruit, embryo sac present inside the ovule. Okay, so see, we have to see in this question, we have to see two things. The first thing is that one plant part should be present inside other one. And the two parts should be from different generations. Okay. So let us see options now. So the first option is saying one within the other pollen grain inside the anther. So pollen grain, when it will fertilize, it will form a new plant. So pollen grain is a new generation. Okay. And anther is belonging to the parent plant. Are you getting me? So there is a parent plant. It has a pollen grain. The parent plant is representing one generation and the pollen grain will fertilize and form a new embryo, isn't it? So this is representing another generation, new generation. So this statement is correct because pollen grain is present inside anther. Anther is a part of stamen. Okay, so it is correct because pollen grain is present inside anther and pollen grain and anther are representing two different generations. Understood? So like that. Now let us see germinated pollen grain with two male gametes. Germinated pollen grain with two male gametes. So what does it say? See, male gametes are present in pollen grain. That's correct. But male gametes and pollen grain, both of them belong to one generation only. Okay. Because pollen grain form male gamete and male gametes fertilize ovum to form new embryo. Okay. So both of them are belonging to same generation. That is why option 2 is incorrect. Okay. Now let us see the option 3. What does it say? It says seed inside fruit. Okay. So seed is formed from embryo and fruit from ovary. Fruit is formed from ovary and seed is formed from ovule. Oh, sorry. Seed is formed from embryo. And both these things are belonging to same generation. Means seed is also a part of new generation and fruit is also a part of new generation, which is formed after fertilization. So even though seed is present inside fruit, both of these are a part of same generation. That is why this is a wrong statement. Moving on to fourth statement. What does it say? It says embryo sac is present inside ovule. See, ovule is present inside ovary and it houses embryo sac. So embryo sac after fertilization will form a new plant which is representing new generation. Okay. So ovule is a part of parent plant. Embryo sac which is present in parent plant will fertilize and form a new plant which is a part of new generation. That is why this statement is correct. Okay, embryo sac is present inside ovule and embryo sac after fertilization will form a new plant. That is why it is belonging to new generation and ovule is present in parent plant. So it is belonging to old generation. Okay, so this statement is correct because embryo sac is present inside ovule and it is showing two generations. Understood? So answer to this question is option number 4. Okay. Moving on to next statement. Pollen grains can be stored for several years in liquid nitrogen having a temperature of -196 degree. So answer to this question is option number 3. This is very simple question and direct bit from your NCERT. Okay. Moving on to next question. Advantage of cleistogamy. Okay. So the question says, what is advantage of cleistogamy? Yes. So question number 103. What is it saying? It is saying advantage of cleistogamy. What is advantage of cleistogamy? It is option number 1, which says no dependence on pollinators. Okay, no dependence on pollinators. Now, what is cleistogamy? Cleistogamy is a process in which what is cleistogamy? Cleistogamy is a process in which there is pollination and fertilization occurring at the bud stage of the flower. So when flower has not opened and it is in the bud stage, at that time only the pollination and fertilization are occurring, and that process is known as cleistogamy. Now, how does this happen? See, what happens is, in the bud stage of the flower, there is a stamen and there is a pistil. Okay. So, what happens is, in the bud, in cleistogamous flowers, the stamen and pistil are present on the same flower. So during the dehiscence of the anther or the stamen, the pollen grains are shed on the pistil and they come in contact with the stigma. Okay. So here the pollination is occurring. Understood? What is happening is, in cleistogamous flowers, the stamen and the pistil are present in the same flower. Okay. So in bud stage, when the anther is when the dehiscence is occurring in the anther, at that time the pollen grains are coming from the anther and coming from the anther and lying on the stigma. This allows contact between the pollen grain and the stigma. Okay, allows the contact between the pollen grain and the stigma, and this is known as pollination. Okay, so in cleistogamous flowers, pollination occurs in the bud stage only and inside the flowers. And there is no cross-pollination. Okay, there is no cross-pollination. Autogamy only occurs. What does autogamy represent? Pollination of the stigma with the pollen grain of its own flower. Okay, understood. So in cleistogamy, there is no requirement of pollinators because the stamen is present in that flower only, and after the dehiscence of the anther, the pollen grains from that flower will go to the stigma of the nearby pistil and allow pollination to occur. So there is no need of pollinators in cleistogamy. Understood? So answer to this question is option number 1. Now let us see question number 104. What is perisperm? Perisperm, my friends, is perisperm is persistent nucellus. Okay, this is very easy question. What is perisperm? It is a direct line from NCERT which says perisperm is persistent nucellus. Okay. So now, see, this is an ovule. Inside ovule, there will be a layer of cell, which is known as nucellus. This nucellus, if it persists after fertilization, it will form perisperm. It has reserve food material. Example for persistent perisperm is peat and black pepper. Okay. Yes. So moving on to next question now. Question 105. Let us see what does it say. Through which cell of embryo sac does the pollen tube enter the embryo sac? Synergids. Okay, pollen tube will enter the embryo sac through synergids. So there are two synergids. Central sac, antipodal cells. Okay, so these are synergids. And this is micropylar end. Okay, so the pollen tube will enter through this micropylar end. At that time, degeneration of synergids occur and the pollen tube enters through degenerated synergids. Okay, so the pollen tube enters the embryo sac through degenerated synergids. Answer to question 105 is option number 3. Okay. 8 nucleate embryo sac is, see, 8 nucleate embryo sac can be monosporic, can be bisporic, can be tetrasporic. So answer to this question is any of these. Okay. Fourth option is the answer to question number 106. Yes. So what does monosporic embryo sac means? Monosporic embryo sac means that it is developing from one megaspore. Okay, it is developing from one megaspore. So, one megaspore mother cell forms 4 megaspores. Out of these 4 megaspores, 3 megaspores, they degenerate, and the one left megaspore will form embryo sac. And this is known as monosporic condition. Okay, this is known as monosporic condition. Understood? In bisporic condition, out of the 4 megaspores which are formed from megaspore mother cell, 2 megaspores develop and form embryos. So this is bispore. Okay. In tetrasporic, all the 4 megaspores will form embryos. Okay. Understood. So, embryo sac can be monosporic, bisporic, tetrasporic. Therefore, answer to this question is option number 4, that is any of these. Understood. So, this was about question 106. Yes, moving on to question 107. What does it say? It says, which of the following is not an attribute of a population? So, the answer to 107 is option number 4. Species interaction refers to mutualism, commensalism, like that. It is species interaction is interaction between two species which can be beneficial to one or other species. Okay, understood? So answer to question 107 is option number 4. Okay, moving on to next question. Which one is true? Commensalism when none of the interacting populations affect each other. Okay. Symbiosis when interaction is useful to both populations. Symbiosis when neither population affects each other. Commensalism when interaction is useful to both populations. Okay. So let us evaluate. Okay. So we have commensalism is interaction is useful to one and other interaction. There is no effect on other organism. And mutualism or symbiosis is the interaction is useful to both the organisms. Okay. So now let us see. Commensalism and none of the interacting population affect each other. This is wrong. Okay. Because one interacting population will be benefited and the other interacting population will have no effect. And that is known as commensalism. Okay. The option 2 is symbiosis. Symbiosis refers to interaction is useful to both the population. Yes, this is correct. Let us see option C. It says symbiosis when neither population affects each other. So this is wrong. Symbiosis is when both the populations of the interaction are useful to each other. Okay. And commensalism refers to interaction is useful to one population of the one population and it does not affect the other population. Okay. So one population is getting benefited and the other population is not getting affected, and this is known as commensalism. Okay. What is it known as? It is known as commensalism. Question 109. What does question 109 say? It says, a biologist studied the population of rats in a barn. He found that the average natality was 250, average mortality was 240, immigration 20, emigration 30. The net increase in population is. See, natality is newborns or number of births. Natality is number of births per 1,000 population. Mortality is number of deaths per 1,000 population. So natality is birth per 1,000. Mortality is death per 1,000. Immigration is people coming in. Immigration is people going out. Okay, so what is net increase? Let us see. See, mortality removes people from population and emigration removes people from population. So let us see how many people are removed. It is 240, that is mortality, plus emigration 30. So this is 270. Okay. And natality includes people in the population and immigration adds people in the population. So immigration and natality add people in the population. So let us see. Natality is 250. Immigration is 20. So net it is 270. Okay, so 270 people are added and 270 people are removed from the population. So net increase will be people added minus people removed, that is 0. So answer to this question is option number 2. Understood? Question 1010. A sedentary sea anemone gets attached to the cell lining of the hermit crab. What type of association is this? This is commensalism. Okay. The interaction between sea animal and hermit crab is commensalism. Understood? Gauss's principle of competitive exclusion states that no two species can occupy the same niche indefinitely for same limiting resources. Larger organisms exclude smaller ones through competition. More abundant species will exclude less abundant species through competition. Competition for the same resources excludes species having different food preferences. Yes. Sorry, there was a glitch. So yes, coming back. So Gauss's principle of competitive exclusion, what does it say? Gauss's principle of competitive exclusion says that two or more species, which are two or more species, which are having same niche, which are having same niche or similar niche, cannot exist indefinitely in the same area, because sooner or later they will come into competition of possession of the niche. Okay, so it says that when two species are having same niche, then both the species cannot exist in same area because ultimately the two species will compete for the same niche. Understood? So what it is saying that when two species are having, species 1 and species 2 is having same niche, it cannot live in one area indefinitely, matlab forever. Okay, indefinitely, because at one point these two species will come into competition, to acquire this niche. Understood? That is why two species with same niche cannot live in area indefinitely, because at one point they will be competing to acquire that. Okay, so answer to question 111 is option number 1, which is saying no two species can occupy the same niche indefinitely for same limiting resources. Okay, understood? So answer is option number 1. Asymptote in logistic growth curve is obtained when k is equal to n. So when k is equal to n, asymptote is obtained. K is carrying capacity, n is number of organisms. Okay. So logistic growth curve is a curve or a graph which says that there will be increase in population of an area until there is carrying capacity is reached. Okay, so logistic growth curve says there will be increase in population of an area, or the rate of growth of population will be increasing until carrying capacity is reached. Understood? What is carrying capacity? Carrying capacity refers to maximum number of organisms, and area can maximum number of organisms that an area can sustain
with available resources. Okay, so maximum number of organisms that an area can sustain with available resources is known as carrying capacity. And when carrying capacity equals the number of organisms in population, then asymptote is reached in logistic growth curve. And what is logistic growth curve? Logistic growth curve is a graph which says that there will be increase in rate of growth of population until carrying capacity is reached. Okay. So logistic growth appears something like this. Curve appears something like this. S shape. Understood? So before moving on to the next question, let me tell you the equation of logistic growth curve, which is very important. You should remember. It is dn by dt equals r into n multiplied by k minus n by n. K. K is carrying capacity, n is number of organisms in population, r is intrinsic rate of growth. Okay, and dn by dt is increase in rate of growth of population. Understood? Moving on to question 113. What does it say? It says, a tall homozygous pea plant upon selfing does not produce a tall heterozygous plant. This is a correct statement. See, tall homozygous means capital T, capital T, capital T, capital. This is self-cross. Okay, it has gametes of one variety only. That is capital T, capital T, capital T, capital T. All these can combine to form only one type of organism, which is capital T, capital T, capital T. Okay. And this is homozygous tall. Understood? So homozygous tall pea plant cannot self and produce homozygous cannot self and produce heterozygous tall. Okay. So when homozygous tall plant is self, it produces homozygous tall plants. It cannot produce heterozygous tall plant. Statement 2. A tall heterozygous pea plant upon selfing does not produce either tall homozygous or dwarf homozygous progeny. So this is wrong. Wait, we'll see. Before saying this is wrong, we'll see. Tall heterozygous pea plant means capital T small t. Okay. Upon selfing means capital T small t crossed with capital T small t. Okay. We have gametes of two varieties here. Capital T and small t. Let us make a Punnett square. Capital T, capital small t, capital T, small t, capital T, capital T, capital T, small t, capital T, small t, capital T, small t, small t. So we will have one capital T, one capital T. We'll have one capital T, one small t, one capital T, small t, and one small t, small t. So we will have a tall heterozygous plant upon selfing can produce tall homozygous, yes, or dwarf homozygous, yes. So this statement is also wrong. So answer to this question will be statement 1 and 2 are wrong. So let us see. So option 4 is the correct answer to question 113. Let us move on to question 114 and see what does it say. Down syndrome, it is a match the following type of question. So Down syndrome, we have an additional 21 chromosome. 21st chromosome is also an autosome. So we have an extra autosome in Down syndrome. So A is 2. In Klinefelter syndrome, we have an extra X chromosome, which is a sex chromosome. So Klinefelter syndrome, we have additional sex chromosome. So A is 1. A is 2, B is 1. And in Turner's, we have 44 plus XO. So we have this in Turner's. And polyploidy can be seen in plants. So A2, B1. C3, D4. So answer is option number 3, A2, B1, C3, D4. Okay. Moving on to 115. Okay. Match the following. What is dominant pod color? Dominant pod color is green. So A1. Recessive pod color yellow. B2. Dominant seed color. What is dominant seed color? Dominant seed color is yellow. So C4, D recessive seed color is green. So A1, B2, D, C4, D3. Okay, so we have third option as the correct answer. 115, option number 3. One one six. Based on Mendel's observation on monohybrid cross, he proposed two laws. Law of dominance, law of segregation. Okay, and based on dihybrid cross, he proposed law of independent assortment. So answer to this question is both option 1. Okay, very simple. Genetics, that is your principles of inheritance and molecular basis of inheritance chapter. Both of them are such chapters, no, that if you properly understand them once in your lifetime, you will never forget. Such beautiful chapters. Yes. So sugar polymer, which decides the type of blood group, is located on the membrane of red blood cell. Plasma membrane of the red blood cell. So option 3 is the correct answer to question 117. Okay, the plasma membrane of the red blood cell has a polysaccharide, which will decide that whether it is going to be A blood group or B blood group, because that polysaccharide will act as A antigen or it will act as B antigen depending on the nature of the polysaccharide. Okay. Moving on to question 117. Sorry, 118. I'm sorry. Yes. Sorry for the glitch. Moving on to question 118. Consider the following Mendelian cross. Capital A, small a, small a, small a. What is the phenotype of offspring? Okay, let us see. Capital A, small a, small a, small a, crossed. So here we have two types of gamete. Capital A, small a. Here we have two types of gamete. Small a, small a. And we will get, let us make a Punnett square. Small a, small a, capital A, small a, capital A, small a, capital A, small a, small a, small a, small a, small a. So we have capital A, small a, capital A, small a, small a, small a, small a, small a. Okay. So these are recessive, that is 50% are recessive and 50% are dominant. Understood? So we have, the answer is 50% are dominant. Option 3 is the answer because first option says 100% recessive, which is an incorrect option. Second option says 75% recessive, which is again incorrect option. Third option says 50% are dominant. This is correct option. Fourth option says 100% dominant. This is incorrect. Because it is incorrect, this is incorrect, and this is incorrect. So answer is third option. Understood? 119. Female produces heterogametes. This is a spelling mistake. Never mind. Female produces heterogametes in which of the following organisms? Insects, man, Drosophila, and birds. See, female of human beings never produce heterogametes. Okay, females produce X chromosome and X chromosomes. In human beings, male produce X chromosome and Y chromosome. Okay, so man is an incorrect option. So answer to this question is female heterogamete. That is, two different kind of gametes are produced by a female in Drosophila. Okay, so Drosophila represents female heterogamete. Understood? Moving to question 120. Which of the following is autosomal dominant? Myotonic dystrophy is autosomal dominant. Okay. You have to remember, you have to by heart this. That what are the different diseases which are autosomal dominant, what are different diseases which are autosomal recessive, what are different diseases which are X-linked dominant and X-linked recessive. See friends, this thing will be asked in your MBBS fourth year also, final year also. So you have to remember this no other option. Yes, so let us read this question. Sorry. Yeah. So due to non-disjunction of chromosomes during spermatogenesis, some sperms carry both 22A plus XY, while others do not carry any sex chromosome. That is 22A plus O means no sex chromosome. And if these sperms fertilize a normal 22AX, what type of genetic disorder appear among these offspring? Okay, so let us see. We have one 22A plus XY sperm, and we have one 22A plus O sperm, and we have an ovum 22A plus X. So if this ovum is fertilized by this, then we get 22A plus XXY, and if this ovum is fertilized by this, we get 22A plus XO. So this gives Turners and this gives Klinefelters. Okay, understood. So answer to this question is option number 1, Klinefelter syndrome and Turner syndrome. Okay. So question 121. Answer to question 121 is option 1. Yes, so let us see question 122 now. What does question 122 say? It says that the genotype of a plant showing dominant, the genotype of a plant showing dominant phenotype can be determined by test cross. What is test cross? Test cross is a cross formed between a dominant phenotype whose genotype is to be determined. We know that it is a dominant phenotype, but we do not know whether it is homozygous or heterozygous. So we want to know the genotype of this dominant phenotype. So to know this genotype, we will cross this dominant phenotype with its recessive parent. Okay, understood, and this cross is known as test cross. Moving on to 123 now. Capital R red antirrhinum is crossed with capital W white one. Offspring RW is obtained, which is pink in color. So this is an example of, come on, it is an example of incomplete dominance because capital R is incompletely dominant over W. That is why capital R and W is pink and not red. Okay. If it was if the capital R allele was completely dominant over W, then the pink would be, then the capital R W would be red in color and not pink in color. Okay. So it shows incomplete dominance. Question 124. In the lac operon model, lactose molecule function as inducer. Let us see. In lac operon model, the lactose molecule functions as inducer, which binds to repressor protein. So see, in lac operon, what happens is, okay, this is a big one. In lac operon, we have an inducer, which is lactose. It binds to repressor protein. So when lactose binds to repressor protein, the repressant protein undergoes a conformational change and this conformational change in the repressor protein see, now this is lactose. It binds to repressor. So let us use the capital words. Lactose binds to repressor protein. Okay. Under repressor proteins undergoes conformational change. Now this conformationally changed repressor cannot bind to operator protein. So this allows RNA polymerase to reach the promoter and perform transcription. This transcription produces mRNA, which is later converted to protein enzymes. Okay, and these enzymes are required for these enzymes help in lactose metabolism. Okay. So this is what happens when lactose is binding to repressor protein. Then the repressor protein will undergo conformational change, and this conformationally changed repressor protein cannot bind to the operator protein. Okay, so what happens is the RNA polymerase can reach the promoter protein, and when the RNA polymerase reaches the promoter protein, it synthesizes an mRNA through transcription. That mRNA is converted to protein enzymes through translation, and these enzymes help in metabolism of lactose. Okay. This happens when lactose is present in environment. So it is an inducer. Induces like operon function. If it is absent, this will not happen because let's see what happens if the lactose is absent. If the lactose is absent, it will not bind to repressor. Repressor will not undergo conformational change and it will bind to operator. So when repressor binds to operator, the RNA polymerase cannot reach promoter. And when RNA polymerase cannot reach promoter, there is no transcription of mRNA which is required for synthesis of protein enzymes. Okay, that is translation. So there is no synthesis of protein enzymes. So what happens? No lactose, repressor bound to operator, RNA polymerase cannot reach promoter, no transcription occurs. Understood? So lactose is an inducer of lactose. So answer is option number 3. Let us see question number 125. 125. Okay. Let us see question number 125. So moving on to question 125. What do we see in question 125? Match list 1 with list 2 and choose the correct answer. So A gene. What does A gene do? A gene produces trans-acetylase protein. Okay. So A gene produces trans-acetylase protein. Option A is 2. Okay. Y gene produces permease. Option B is 3. C gene. C is gene I. I'm sorry. Yes. Gene I produces repressor protein. So option C is 4. Option C is 4. Gene Z produces beta-galactosidase. Okay, gene Z produces beta-galactosidase. So option D is 1. So A is 2, B is 3, C is 4, and D is 1. Okay, so we have option 3 as the correct answer. Understood? We have option 3 as the correct. So answer to question 125 is option 3. Okay, which says gene A synthesizes trans-acetylase Gene Y synthesizes permease and gene I synthesizes repressor protein and gene Z synthesizes beta galactosidase. Okay, understood. So moving on to next question now. Question 126. So what is the answer to question number 126? The answer to question number 126 is polymerase, it breaks DNA into fragments. Okay, this is wrong. Polymerase synthesizes DNA. Nuclease, it separates two strands of DNA. Wrong. Topoisomerase does that. Nuclease breaks DNA. Exonuclease makes cut at specific positions in DNA. No, that is endonuclease. Okay, so these three options are wrong. Let us see what does the fourth option say. Ligase joins the two DNA molecules. Correct. This is correct. So answer to 126 is option number 4. Choose the correct statement. So question number 127. Choose the correct statement. So we have 6 statements in total. Let us see. Choose the correct statement. Binding of mRNA to ribosome. Binding of mRNA to binding of mRNA to ribosome is loose and reversible. It's wrong. It is wrong. Okay. I'm sorry, just a second. Let us choose the correct. Binding of mRNA to ribosome is loose and reversible. Yes, it's correct. Loose and reversible. So after protein synthesis, the mRNA will detach from the ribosome. So it's reversible also and it's loose. Moving on to second statement. It says the ribosome, which is composed of tRNA and proteins, consists of small and large subunit. The ribosome, which is composed of tRNA and proteins. Ribosome is not composed of tRNA and proteins. It's wrong statement. Each of the 20 amino acids have minimum of one specific tRNA molecule. This is correct. Each of the 20 amino acids have at least one specific tRNA molecule, and this is a correct statement. Okay, understood. Now let us move on to question statement number 4. It says methylated tRNA binds to the first codon on the mRNA, that is the start codon. Yes, this is correct option. Methylated tRNA only binds to the start codon during translation. What is start codon? Start codon is known as AUG. Okay. So this is also correct. So we have 1, 3, and 4 statement as correct statements. Let us see fifth. The second tRNA binds to second codon so that their attached monosaccharides are next to each other. No. See, the second tRNA binds to the second codon. It is a correct statement. But so that their attached amino acids are not monosaccharides. So that their attached amino acids are next to each other. Understood? So this is wrong statement because instead of monosaccharide, here amino acid should be there. The option number, the statement 4 is the newly formed polypeptide may undergo post-transcriptional modification. The newly formed polypeptide may undergo post-transcription. The newly formed polypeptide will undergo post-translational modification and not post-transcriptional modification. Therefore, this statement is also wrong. So answer will be option number 1, that is 1, 3, and 4 are correct. Okay, understood. Moving on to question 128. What is the role of RNA polymerase 3 in process of transcription in eukaryotes? RNA polymerase 3 synthesizes tRNA, 5S rRNA, and small nuclear RNA. So option number 3 is the correct answer to question 128. Understood? How many different proteins does the ribosome consist of? Ribosome consists of 80 different proteins. Okay, ribosome consists of 80 different proteins. This is an NCERT statement and it is the answer to the question 133. sorry, 129. Question 130. Let us see what does it say. Question 130. VNTR are useful in DNA profiling because they are hypervariable. Yes, they are hypervariable. The name itself says that. What is the name? Variable number of tandem repeats. It says that VNTRs are hypervariable. They are inherited. Yes. They synthesize constitutive enzymes. This is wrong. What are constitutive enzymes? Constitutive enzymes are enzymes which are constantly active 24 hours in our body. So these enzymes are synthesized in the body but not, these enzymes are synthesized in the body. These enzymes are synthesized in the body during translation. Okay, but not through VNTRs. Okay, VNTRs, variable number of tandem repeats, do not synthesize constitutive enzymes, but they are hypervariable and they are inherited. So answer to question 130 is option 1, sorry, statement 1 and statement 2 are correct. That is second option. Okay. Okay. Understood. Yes, so question number 131. In RNA splicing, exons are removed and introns are joined together. RNA splicing occurs before translation, and it is removal of introns and joining of exons. Okay, so RNA splicing is removal of introns, joining of exons. That is option 2 is correct. Okay. Question 132. The Okazaki fragments in DNA chain growth result in transcription, polymerize 3' to 5' direction and form replication for proof semi-conservative nature of DNA replication. Polymerize in 5' to 3' explain 3' to 5' direction of DNA replication. So answer to question 132 is option number 4. What are Okazaki fragments? Okazaki fragments are fragments of newly replicated DNA synthesized on 5' to 3' template strand. Okay. So, because the DNA polymerase can polymerize DNA only in 3' to 5' direction, that is why the DNA molecule which is synthesized on the strand of 5, see, let me simplify it for you. So, this is DNA, open DNA strands, okay, or uncoiled DNA strands. This is 5' to 3' strand and this is 3' to 5' strand. Okay, so now for replication to occur, the DNA polymerase will replicate DNA in the direction of 3' to 5' like this on this strand. Okay, template strand. But on this strand, that is the strand that which is 5' to 3' direction, the DNA replication cannot occur. Okay, why? Because the DNA polymerase can replicate only in 3' to 5' direction, but not in 5' to 3' direction. Understood? The DNA polymerase can only replicate in 3' to 5' direction, but not in 5' to 3' direction. So, what do? We perform replication we perform replication, and this replication of DNA occurs through small fragments of DNA. So small fragments of DNA are synthesized, or these fragments are known as Okazaki fragments. So small Okazaki fragments are small fragments of newly synthesized DNA on the 5' to 3' strand of the parent DNA. Okay, why? Because DNA polymerase can synthesize new DNA only on the 3' to 5' strand and not on the 5' to 3' strand. Understood? Moving on to question 133. Whose experiments cracked the DNA and discovered unequivocally that a genetic code is a triplet? Okay, Nirenberg and Matai are the people who discovered that genetic code is a triplet. Understood? Yes. Which of the following is the sequence on corresponding coding strand if the sequence of mRNA is formed as follows? Okay. The sequence of mRNA is formed as follows. See, before that, I want to tell you one more thing. The Okazaki fragments which are formed, no? So this was DNA and this was our rep. So the newly synthesized DNA, which is synthesized on the 3' to 5' direction of the parent DNA, is known as leading strand, and the newly synthesized DNA, which is synthesized on the 5' to 3' direction of the parent strand, is known as lagging strand. Okay, because it is synthesized in form of Okazaki fragments. Okay, so this is known as lagging strand and this is known as leading strand. Understood? Now moving on to question 134. I'm sorry. Yes. So this is the question says that we have given you a coding strand and you have to synthesize mRNA from this. Okay. So the sequence of nitrogen base in coding strand and in mRNA are same. Okay, except that in place of U in mRNA, we have thymine in coding strand. Okay, so the sequence of coding strand and mRNA are same, but we have uracil in mRNA and thymine in coding strand. So this question says that we have given you mRNA sequence. You have to synthesize coding strand from this. So what I told you, coding strand is similar to mRNA. Yes, so the whole strand will be similar to this, but coding strand will have thymine in place of uracil. Okay, because this is an RNA, it has uracil. The coding strand is DNA, it will have thymine. So what we have to do is we have to replace uracil with thymine. All these uracils will be replaced by thymine. Okay, so we have ATCG, 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 ATCG. ATCG, ATCG, ATCG 5' to 3'. So option number 4 is the correct answer to question 134. Okay, so what was question 134 saying? It was saying that I am giving you an mRNA. You have to synthesize coding strand from that. Okay, and coding strand of DNA is similar to mRNA. Okay, except that in the DNA, there is thymine in place of uracil. Understood? Question 135. Oh God, this is the easiest question of whole paper. It says that which of the following is start codon? AUG is start codon. Okay. UAA is stop codon. You should remember that. UAA is stop codon. Moving on to question 136. Endosperm is formed during double fertilization by Endosperm is formed during double fertilization by two polar nuclei. Fusion of two polar nuclei with male gamete. Okay. Understood? So answer is option number 1. A simple question. Unisexuality of flowers prevents autogamy and not gytonogamy. Okay. So autogamy refers to pollination of pistil with pollen grains of stamen of the same flower. Okay. Autogamy refers to pollination of pistil with pollen grains of stamen of the same flower. So if the flowers are unisexual, means one flower has either a stamen or pistil. Okay. So a flower will have either stamen or pistil. It will not have both stamen and pistil. So such flowers will prevent autogamy. Okay. Such flowers will prevent autogamy. Okay, so that is autogamy. So when one flower has only stamen or pistil, then self-pollination cannot occur. Autogamy cannot occur. So unisexuality prevents autogamy. Okay, but not gytonogamy. What is gytonogamy? Gytonogamy is pollination of pistil with pollen grains of stamen of the flower of the same plant. Another flower of the same plant. So what is gytonogamy? See, this is a plant. It has two flowers. Okay, this flower has pistil. This flower has stamen. Okay, so the pollen grain of this flower are going on the pistil of this flower. Okay, and this is the same plant. So when there is pollination of pistil with this pollen grain of the stamen of the same plant, okay, but from the another flower, then that is known as gytonogamy. Okay, gytonogamy. What is gytonogamy? Gytonogamy is pollination of pistil with pollen grain of another stamen from another flower of the same plant. Okay. So such a pollination cannot be stopped by unisexual flowers. Okay, because if the plant has both male and female flowers, then gytonogamy will occur. So unisexual flowers can prevent autogamy, but not gytonogamy. Now xenogamy is also given. So let me tell you what is xenogamy. Xenogamy is pollination of pistil with pollen grains from another flower of another plant. Okay. So unisexuality prevents autogamy, but not gytonogamy and xenogamy. Okay. So answer is option 3. Now moving on to 138 question number. 138 question number says wind pollinated flowers are. Wind pollinated flowers are large. Sorry, wind pollinated flowers are small. Large, small. They're small because they don't have to attract an insect and look beautiful so that an insect sees the flower and feels, wow, such a beautiful flower, I'll go to this flower. Such a thing should not be done by the wind pollinated flowers. So they are small. Okay. They're multiple in number and they produce dry pollen grains which are lightweighted. Okay, so if pollen grains are dry and lightweighted, they can be easily carried by the wind. Okay, and they are multiple because it increases the chances of pollen grain to land on the flowers. Understood? So wind pollinated are small, multiple, produce small, dry, and lightweighted pollen grains. Okay. So that was about question 138. The answer is option number 2. Moving on to question 139. What does question 139 say? Question 139 says, which one of the following is most appropriately defined? Host is an organism which provides food to another organism. Okay, let's see. Amensalism is a relationship in which one species is benefited, whereas another is unaffected. No, this type of relationship is commensalism. Predator is organism that catches and kills other organism for food. This is correct. Yes, let us see. Parasite is organism which always lies inside the body or other organism and may kill it. Always lies inside the body is wrong. Okay. Parasite can lie on the body also. So this is a wrong statement. Okay, predator is organism that catches and kills the organism for food. It is correct. Okay, example lion. Amensalism, it was wrong only. Host is an organism which provides food to another organism. Not always. A host is an organism which houses another organism. Okay, the host can provide food and cannot provide food also. So host is an organism which houses other organism, but it can and cannot provide food. Okay. So answer to this question is 130. 140. In growing population of a country, pre-reproductive individuals are more than reproductive individuals. The example is India. Okay. Pre-reproductive individuals are more than reproductive individuals in a growing country. Example of such a country is India. Option 140, question 141. When does growth rate of a population of a following the logistic model equals 0? See, dn by dt is equal to Rn into 1 minus n by k. Okay, see. Look at this very carefully. Okay, see. This is the logistic growth. It says that rate of growth of a population will increase until and unless carrying capacity is reached. What is carrying capacity? Carrying capacity refers to maximum number of organisms an environment can sustain with its available resources. Okay. So now let us see this dn by dt. So it is saying when will this dn by dt will be 0. So for this dn by dt to be 0, if n is equal to k, then what will happen? See, n is equal to k, so it will be r into n by 1 minus n by n, let's say. So this cancels out. We have r into n equals into 1 minus 1, which is 0. Anything which is r into n multiplied by 0, which is 0. Okay. So logistic model equals 0 when n by k is, when n by k is 1. When n by k is 1 or when n is equal to k. Understood? So answer to question 141 is option number 3. Phenylketonuria is recessive hereditary disease caused by body's failure to oxidize an amino acid phenylalanine to tyrosine because of a defective enzyme. Yes, this is a correct statement. 142 assertion is correct. Okay. It results the presence of phenylalanine acid in urine. Okay. So the answer, see what it is saying, that phenylketonuria is a hereditary disease, autosomal recessive disease, which is occurring due to failure of body to convert phenylalanine to tyrosine due to presence of a defective enzyme. Okay, this is correct. Reason, it results in the presence of phenylalanine acid in urine. Okay. So let us see. The options are if both assertion and reason are true and there is a reason is the correct explanation to assertion. Okay, both assertion and reason are true, but the reason is not the correct explanation to assertion. Next, if assertion is true, the reason is false. Both assertion and reason are false. Okay, so the answer to this question would is option number 2. That is, if both an assertion and reason are correct statements, but the reason is not the correct explanation to assertion. Okay, so reason says there is release of or secretion of phenylalanine acid into urine. Obviously, if phenylalanine cannot be converted to tyrosine in body, phenylalanine will be secreted in urine. Okay, So it is a correct statement. Both of them are correct statement, but reason is not the correct explanation to assertion. So option number 2 is the correct answer. In your PDF, option number 1 might be given. So that's a printing mistake. Never mind it. Answer is option number 2. 143. The physical association of chromosome is called genes on. The physical association of chromosome is called genes on linkage. Okay, so physical association of genes on chromosome is called linkage. So the question is this is some grammatical mistake. Okay, but the question is the physical association of genes on chromosome is called linkage. A child's blood group is O. The parents' blood groups cannot be A and B, A and A, B and O, B and O. Okay. So child's blood group is O. So for child's blood group to be O, it has to be I. Okay, let's say it has to be I. So for blood group, we have three alleles, IA, IB, and I. So these two alleles are co-dominant and these two alleles are dominant on this one. Okay. So if the parents' blood group are A and B, it will be IA, IO, IB, IO. So it can give IO. Can give IO, IO. So if parents' blood group is A and B, the parents can have a genotype of IA, IO, and IB, IO. Okay, and this IO and IO will combine to form IO, IO, and it will give O blood group. So yes, option 1 cannot be the correct answer because if parents' blood group are A and B, the child can be O blood group. Okay, moving on to A and A. So if child blood group is, if the parents' blood group is A, and it can be IAIO, IAIO. If IAIO are there, then this IO and this IO will combine to form IO. Then the child will have O blood group. So if parents' blood groups are A and A, the child can have O blood group. No worries. Let us see option number 3. There's A, B, and O. So if the parent has A, B blood group, then it will be IA, IB, and IO, IO. So in this scenario, the alleles will be IA, IB, IO, IO. So the offsprings will be IA, IO, IA, IO, IB, IO, and IB, IO. So it will be, this will be A, this will be A, this will be B, this will be B. So if parents' blood group is A, B, and O, the child cannot be O blood groups. So answer to question 144 is option number 3, that is A, B, and O. But let us see option 4 also. The parents' blood group is AB, means it can have a genotype of IB and IO, and the parents' blood group is O. So it will have a genotype of IO, IO. Okay. So it will form IB, IO, IB, IO, 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 IO. So this is, it can have B blood group and it can have O blood group. Okay, so children of a parent with B blood group and O blood group will be B blood group or O blood group. So answer to this question is option number C, which says AB blood group and O blood group parents cannot have a child with O blood. Okay. Question 145. If both parents are carriers for thalassemia, it is an autosomal recessive disorder. What are the chances of pregnancy resulting in affected child? See, thalassemia. Okay, so let us take, what should we take? Let us say take T only as the gene for thalassemia. Okay, it is autosomal, so T only will take. Capital T is dominant, small t is recessive, is an autosomal recessive, so both are carriers. That means both the parents have one small t. Okay. So now these are the parents. If we cross them, we will get capital T, capital T. Okay. See, this is capital T. Okay. So this is H gene for thalassemia. Let us suppose. Okay, this has two alleles. One is normal allele, one is thalassemic, the small h. Okay. So both the parents are carriers. That means both the parents have one thalassemic allele. So the father is a capital H small h and the mother is capital H small h. Okay, if we cross both of them, we will get gametes of capital H small h, capital H small h, and children we will get of capital H small h, capital H small h. So H, capital H small h, capital H small h, small h, small h. Okay. So we'll be having capital H, capital H, capital H small h, capital H small h, small h, small h. Okay. So these are normal carriers, thalassemia. So there is 25% of chance. Means 1 out of 4 child can be thalassemia. 25% chance. Okay, so answer to question 145 is option number 1. I'm sorry, I have to erase all this. Just a minute, bear with me. The teacher is a slow teacher. So question 146. Okay. If okay, so let us see. Assertion. If it is metabolic, physiological, environmental conditions that regulate the expression of gene. Reason. If bacteria do not have lactose, they would no longer require synthesis of enzymes beta-galactosidase. Okay. So it is saying that metabolic, physiologic and environmental conditions affect regulation of genes. Yes, it is a correct statement. And reason it is giving that in a bacteria, if there is no lactose, then lactoperon won't work and beta-galactosidase gene won't be required for breaking down lactose. So lactose is an environmental condition which is regulating the expression of gene which synthesizes beta-galactosidase enzyme. Yes, so reason is correct. Both A and R are true and R is the correct explanation to A. Okay, understood? Both A and R are true and reason is the correct explanation to assertion. Okay. So answer is both A and R are true and R is the correct explanation to A. That is option number 1. 1 is the correct answer. Okay. Question 147. What does it say? Which scientist conducted experiment with 32 phosphorus and 35S labeled pages for demonstrating DNA is the genetic material? So which scientist? Yes, Hershey and Chase. The famous experiment of Hershey and Chase indicated that DNA is the genetic material. What did they do? They put protein and radio-labeled protein and radio-labeled DNA into bacteriophage and allowed the bacteriophage to replicate. After replication, they saw that only DNA is passed from one generation to next generation, but proteins is not passed. Okay. So this indicated that DNA is the genetic material and not proteins. Understood? So answer is AD Hershey and J. Chase. Great people. From the following, identify the correct combination of salient features. Salient features of genetic code. So the correct combination is the genetic code is degenerate. Yes, it is degenerate. It is non-overlapping and it is non-ambiguous. Okay, so let us see. The genetic code is degenerate. It means the genetic code is degenerate. It means that the multiple codons can code for one amino acid. Okay, the genetic code is non-overlapping, means during the process of synthesis of protein on mRNA, the genetic code is read in a sequence, one after other. There is no overlapping in reading of genetic code. Okay, understood? So it is non-overlapping and it is non-ambiguous. What does the non-ambiguous say? It says that each codon codes only for one amino acid. But one amino acid can be coded by multiple codons. Okay. So this is a very confusing statement, but you have to remember, okay, what does degenerate mean? Degenerate means that one amino acid can be coded by multiple codons. Okay, so let's suppose methionine is an amino acid. So methionine can be coded by not one, not two, but three codons also. But when the genetic code is non-ambiguous, it says that when the genetic code is non-ambiguous, it says that one codon will code only for one amino acid. So you have to remember, I know this is confusion. You have to remember, okay, degenerate, one amino acid coded by multiple codons. Non-ambiguous, one codon coding for only one amino acid. Okay, so this is confusing, but you should not get confused. Okay, practice multiple times, it will come. Understood? So answer is option number 4. See, genetic, why other options are wrong? Let us see. See, it is universal, yes, it is not overlapping. It is not overlapping. It is not ambiguous. So other options are wrong. Okay. Yes, one turn of helix in B form of DNA is approximately. What it is saying, this is one turn of DNA helix. This one turn is 0.34. Sorry, this is 3.4 nanometer. Okay, so answer is third option. See. Okay, so these are base pairs. So between these base pairs, we have 0.34 nanometer of distance, and this one coil will have 3.4 nanometer of distance. Understood? So answer to question 149 is third option, that is 3.4 nanometer distance of one helix of DNA. Okay. Now question 140. Which of the following is not a cloning vector? Now see. BS, YAC, and PBR 322 are cloning vectors. What is a vector? A vector is an organism or material which takes the DNA or a part of genetic material from one organism to other organism. So it's a vector. It's transporting genetic material between two organisms. Okay. So vector is an organism which transports genetic material from one organism to another organism. Okay. So now here, probe is not a cloning vector. Probe is a oligonucleotide sequence of DNA. Probe is a oligonucleotide which is used for cloning DNA at the start. At the start of cloning DNA, we use a oligonucleotide sequence of DNA, which is known as probe, DNA probe. Okay, so BAC stands for bacterial artificial chromosome and YAC stands for yeast artificial chromosome. Both of them are vectors. PBR 322 is a famous vector. It has a nice structure like this. Okay, that will be taught to you in zoology by your Sakshi ma'am. So listen to her carefully. Till then, If you have any doubt on this paper, do tell me in comments. I'm there to resolve them. See you in the next video.